Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about the next biomolecule, that is nucleic acid. So nucleic acids are the polymers called polynucleotides. So what are those nucleic acids? DNA and RNA, deoxyribonucleic acid and ribonucleic acids. So each polynucleotide is made up of nucleotides. So what are these nucleotides? They consist of nitrogen bases, pentose sugar as well as phosphate group. These three we are calling them as nucleotides. So the portion of the nucleotide without the phosphate group, we are calling it as nucleoside. So that means nucleoside contains only nitrogenous base and sugar. Okay, so let us see first nitrogen bases. There are two types of the nitrogen bases, pyrimidines and purines. Pyrimidines, these are having the singles, that is 6-membered ring structure. These are of three types: cytosine, thymine and uracil. Uracil present in the RNA. Purines, adenine and guanine. These two comes in the purines, which have a 9-membered ring structure. In DNA, the sugar is deoxyribose, but both are pentose only. So deoxyribose sugar at the base you can see, and RNA will have the ribose sugar. Okay, so sugar molecule is different in RNA and the DNA, so that's why DNA and RNA the name suggests. Okay, so nucleoside is phosphate group. DNA has a double helical structure with anti-parallel strands. So structure was proposed by Watson and Crick. Okay, so here you can see in the diagram the first three diagrams, which are nothing but the pyrimidines. Next to 9-membered rings, 9-membered structure, that is purines. Last two, they are the sugars. Okay, so two types of the sugars, and how many nitrogen bases? It complete 5. Pyrimidines 3 and purines are 2. Okay, uracil is present in the place of thymidine in the RNA. So here you can see the structure. So here this is a nucleotide. The backbone is forming because of the sugar and the phosphate, whereas at the center the two strands, here the two strands are binded together with the help of nitrogen bases. So adenine always binds with the thymidine with the help of 2 hydrogen bonds, and guanine always binds with the cytosine with the help of 3 hydrogen bonds. They will give the stability, they will form, but backbone is forming by the phosphate as well as the sugar molecule. So here the helical structure, double helical structure you can see with the 2 strands. And what is the width of this? So 20 angstroms or 2 nanometer, and per each turn it contains 11 base pairs. Okay, that is adenine, guanine, these are the base pairs, right? Adenine, thymidine is one base pair, like that 11 are present. DNA molecule has 2 polypeptide chains spirally around. Already we learned double helical structure. So they are anti-parallel, that is they are from 5' to the 3', and other strand 3' to the 5'. Oppositely, anti-parallel strands are present. The nitrogen bases in DNA pair up and form hydrogen bonds. Adenine always bind with thymine, and guanine always bind with cytosine with hydrogen bonds, 2 and the 3 hydrogen bonds for the stability. At first carbon of the sugar is pentose sugar. At the first position nitrogen bond bases will bind, and at the third and the fourth position, sorry, it is fifth actually, fifth position of the sugar, the phosphate groups are present. So that's why the 5' and 3' regions. Okay, so what are the bonds present? Let us see. Bonds between bases are hydrogen bonds, right? Bonds between sugar and the phosphoric acid is present, right, at the third and fifth position. So that bond is phosphodiester bond. And bond, so we know Watson and Crick proposed the double helical structure based on two things: Chargaff's rule as well as Frederick Xtrek crystallography. Chargaff's rule, which states purines, those are A, G, adenine, guanine, and pyrimidine C, T, are equal to the 1. Okay, so there are 3 types of the DNA: B form, A form and Z form. B form and A form, they are right-handed, as well as major grooves are present, whereas the Z form, it is left-handed and they are absent, major grooves are absent. And how many base pairs are present per turn in B form? There are 10.5. This is a common type. 10.5 are present. In A form, 11 base pairs are present per turn. Z form 12 are present. So here you can see the structures and also the different base tails and helix rise also you can see here. So next RNA structure. Another nucleic acid, it contains single stranded structure, helps in the protein synthesis, and it is not a genetic material. Only in few virus it acts as genetic material. And what is the sugar? R stands for ribose, right? Ribose sugar. Nitrogen bases are adenine, uracil, cytosine and guanine. So here thymidine is absent. In the place of thymidine, uracil is present. So here you can see the structure. Single stranded one is the RNA and double stranded one is the DNA. But base pairs, how they are binding, adenine will bind with the uracil in the RNA. So remaining all mostly they are same. Sugar is also pentose sugar. So this is the structure, and here the mutation rate is higher in the single stranded RNA. It will show the structural changes it will undergo. It is unstable structure compared with the DNA. So this is about the DNA and RNA. Nucleic acids, the DNA act as a genetic material, whereas the RNA is not acting as genetic material. It helps in the protein synthesis. It acts as a genetic material only in the few virus types. Thank you for watching. Please subscribe my channel. Thank you everyone.